Let's discuss the histology of microcirculation. So we are entering the arterioles. The arterioles, approximately 1 millimeter in diameter, have the wall made of like 2 to 3 layers of smooth muscle cells, 2 to 4, or let's say, layers of smooth muscle cells that can efficiently change diameter. The branches of the arterioles are called metarterioles because they are behind, like meta-arterioles. And before the metarterioles split into capillaries, they are spiral accumulations of vascular smooth muscle cells, or cells called pericytes, that form precapillary sphincters. That can close or open the capillaries. The capillaries are forming anastomosic networks, and they change from arterial capillaries into venous capillaries, and the confluence of these venous capillaries forms the postcapillary venules and the venules. Venous capillaries and post-capillary venules of various size drain the blood away. Now, there are usually also shortcuts between the arterial and the venous part of the circulation. They could be opened or closed because they also have sphincters, smooth muscle sphincters. And they are called bypass vessels or shunts, shortcuts from the arterial to the venous system. With this architecture, individual organs of our body, like skin, muscles, intestines, etc., might regulate the volume of blood flowing through the microcirculation. It's important to understand that the overall capacity of the microcirculation exceeds the real volume of circulating blood. So if all the precapillary sphincters would relax at once everywhere, we would literally bleed out into our own microcirculation, because there would be no enough blood pressure, not enough blood to perfuse our vital organs. So a sensitive regulation of the precapillary sphincters affects the distribution of the blood in the human body. Let's have a look on capillaries. There are 3 types of capillaries in the human body. The first type has endothelium endothelial cells that are closely attached to each other, leaving no gaps. Moreover, the cells have intercellular tight junctions in between, and they are surrounded by a continuous basal lamina. So we got endothelium, we got tight junctions between the endothelial cells, the tight junctions are sealing the spaces among the cells, so no molecules can pass through the extracellular space. The tight junctions are preventing the paracellular transport, so all the substances that are permitted to cross the wall of the capillary have to pass selectively through the endothelial cells. There is a continuous basal lamina here. This type of capillaries is called continuous. Continuous type. And it is the type with the lowest permeability. It occurs in lungs. It occurs in brain and spinal cord. In lung, it's part of the blood-air barrier. In brain and spinal cord, it's part of the blood-brain barrier. So the blood-air barrier, and here the blood-brain barrier. It also occurs in muscles and in exocrine glands. The second type has gaps between the microscopic gaps between the endothelial cells, and also the cytoplasm is interrupted by microscopic canals that permit small molecules to pass through more easily. The basal lamina has small openings as well. So we got these openings among the endothelial cells, fenestrations. Fenestra is Latin term for window. So there are these fenestrations, which makes these capillaries more permeable. We call this type fenestrated. Fenestrated capillaries. They are more permeable. They occur in kidney, being part of the filtration barrier where the primary urine is filtered from the blood plasma. They are in the intestines, in the intestinal villi. Through this wall of fenestrated capillaries, some nutrients are allowed to pass into the bloodstream. They are in the choroid plexus, through which the cerebrospinal fluid is filtered into the brain ventricles, and they are in the endocrine glands, where through these fenestrations the hormones are permitted to pass into the bloodstream. The third type has really huge gaps among the endothelial cells. They could be irregular, really dilated, and the basal lamina is very often reduced to fragments with really large openings. So there's a discontinuous basal lamina, and there are large gaps. And fenestrations. This type is called blood sinusoids, and while the typical diameter of the continuous type is like 7 to 10 micrometers, so some red blood cells might even be compressed to pass through, the diameter of blood sinusoids might be larger, like 20 to 30 micrometers. They occur in the bone marrow, in the hematopoietic bone marrow. These large gaps permit the foreign blood elements to enter the bloodstream. They are in the liver, liver sinusoids among the hepatocytes, among the liver cells, and the blood sinusoids are also in the red splenic pulp. They even let the red blood cells to pass through to enter the labyrinth of the red splenic pulp cords, called Billroth's cords, where the elderly red blood cells are getting stuck and phagocytized because of the increased fragility of their cell membrane. They are very slow. They cannot be deformed to re-enter the venules of the pulp. So it's part of a filter for the red blood cells. So after the microcirculation, the blood returns via the venules and veins. So let's compare the histology of a vein with the histology of the arteries we already discussed. So what are the major differences? There's also intima, media, and adventitia like in the arteries, but the intima makes folds called valves that are covered by endothelium and supported by the subendothelial connective tissue. So this is endothelium, subendothelial connective tissue. Here we have the valves, one of the leaflet. Then the tunica media contains vascular smooth muscle, which is similar to arteries, but in this case the bundles of the smooth muscle cells are surrounded by a significantly more or greater fraction of connective tissue. And the smooth muscle cells and the bundles can be found also in the adventitia, and sometimes even as longitudinally arranged smooth muscle cells. This does not happen in arteries. And we got much more collagen here. Collagen and glycosaminoglycan. So the quality of the vascular wall strongly depends on the quality of the connective tissue. And in the adventitia we also have the vasa vasorum and the nervi vasorum. So here is a spiral spiral, smooth muscle bundles, but here are even longitudinal smooth muscle bundles, rich collagen connective tissue, collagenous. So the veins are usually less muscular than the arteries, but with more collagen. And here are the nervi, vasorum, and the vasa vasorum. So here is the intima, the media. The transition between the media and adventitia is sometimes not well expressed, and the thickest layer in veins is usually the adventitia, not the media like in arteries. Of course, there are many veins in the human body, and the veins are histologically highly variable. For example, the veins of the lower extremities are really, like the saphenous vein, are really muscular. They contain a significant fraction of smooth muscle. They carry the blood against the gravity.